Okay, well, welcome back, everybody. We've just seen a nice lecture illustrating all the genetic aspects that can play a role in small and fragmented populations, things you need to consider when you're working in the field of conservation. And in this lecture, I want to go back a little bit more to the basis, and I want to try and illustrate how to measure this genetic diversity we have talked about already for almost a day and a half now. First, what I'd like to do is we have talked about diversity on different levels, and you can use molecular techniques, genetic techniques, to study it on these three different levels as well. So you can study it on the ecosystem level, on the species level, or even on the level within a particular species. But the first step is you need your source of information, which is the DNA. Different sources, many different sources of DNA. My own work is mainly focused on lions, so I will include some pictures of lions and some examples of lions in here as well. So you can see a blood sample and the lion. The lion up there was a lion that was sedated to get a GPS collar, and we got a little tissue sample from the ear. And that is a very good source of DNA. The DNA you isolate from this material has a high quality. It's not fragmented, it's nice long pieces, and it's also a high quantity of DNA. But it's not that easy to collect blood or tissue samples of mainly endangered species. So you want to look at other sources of DNA as well. And what you can do is you can also collect DNA from non-invasively collected samples, like the SCAT sample you see up there on the screen as well. And although the quality and the quantity of the DNA is less than in blood and tissue samples, it's still a very good and very convenient source of DNA. Another source would be, for instance, using natural history collections. We sampled quite a lot of lion skulls. We took small fragments from the nose, the bone in the nasal cavity, which can also yield you DNA. And some techniques that are very interesting and upcoming now is with the new sequencing techniques I will talk about later in this presentation. You can also isolate DNA and study DNA from sources like a few droplets of water. You don't even need to find the species you're interested in, but if the species is there from fecal matter or cells they shed in the water, you can still pick up the traces of the DNA, and you can study a species even without actively handling it. So DNA, this is the structure was discovered by Watson and Crick in the 50s. They got the Nobel Prize for it about 10 years later. And what they discovered is it's a double-stranded helix, and the genetic information which is in there is coded by base pairs. And these base pairs, they have 4 different forms. We have A, T, C, and G. They're always combined like that, A with T and C with G. And the sequence of these base pairs, they basically encode the information which is in the DNA. So a big part of the DNA, or an important part of the DNA, contains of genes, regions that yield a protein product. But an even bigger part, in the case of humans, even 98%, the DNA is actually not coding for genes, for protein products, but it's non-coding DNA. And this non-coding DNA can contain all kinds of regions. It can contain regulatory regions that play a role in how much of the protein product of these different genes are produced. They can play a structural role, and it's actually a pretty big part of the DNA which we don't really know what it does. This can be copies of genes that are not functional anymore because they have gained certain mutations. It can also be parts of the DNA we don't really know what it does, so this is what we call junk DNA. So the DNA, the biggest part of the DNA is located in the nucleus of the cell, and we also find smaller parts of DNA which are not in the nucleus, but they're in small organelles called mitochondria. Those are small organelles that play a role producing the energy which is needed for the cell. And those organelles also contain DNA. If you would study plants, for instance, you would also find DNA in another type of organelles called chloroplasts, which we only find in plant cells. But for here now, we're going to focus on an animal cell. And if we first take a look at the nuclear DNA, and we zoom into this cell nucleus and look how the DNA is organized in there, we can see that the DNA is organized in chromosomes, and the chromosomes, you have always two copies, one from your mother, one from your father, and so that is why if there is in a certain chromosome there is a specific gene, this gene occurs in two alleles, one allele from the mother, one allele from the father. The only exception to that can be the sex chromosomes, which are in the case of mammals is X and Y chromosome. For instance, males have one X and one Y chromosome, though there it's slightly different. But for all the other chromosomes, we find these two alleles. Now, if we're going to look at this other source of DNA, we look at the mitochondrial DNA. And this mitochondrial DNA is doing something a little bit differently. It's not organized in chromosomes as we have seen in the nucleus, but it's a circular molecule. And you only have one allele, which is kind of interesting. So you don't have these two alleles where you can compare and look at allelic diversity or heterozygosity. You have only one single allele. And another thing that is important is that it's only maternally inherited. So all the mitochondrial DNA you contain is from your mother. The father does not play a role in this. So if we want to look at the sources of genetic diversity, we've talked quickly already about some things, about mutations and about selection. Now, I want to first take a look at small-scale changes in the DNA. For instance, a point mutation, and it's shown up on the screen on the DNA strand. On top there is a C, and the DNA strand on the bottom contains a T instead of the C. So this is a point mutation. It's the change on the level of a single base pair. And of course, a single base pair can also be deleted, or for instance duplicated, or can be inserted. So that's the changes in the DNA on the level of a single base pair. So to illustrate it a little bit further, I copy-pasted a part of our Lion data set we use, and I know it's very small, but basically what you see, this is a part of the mitochondrial DNA, and every row is a single individual. And we compare the DNA sequences of all these individuals by aligning them and putting them underneath each other. And if we want to see what the differences are in these, what are the variable positions, what are the point mutations, you can show it like this. So you see that the point mutations are highlighted now. There's a long row of A's and C's. So we know that this is a variable position. Some individuals have their one base pair, other individuals have their another base pair. So these are the changes on the single base pair level. But you also have sources of genetic diversity on a larger scale, on, for instance, a chromosomal level. So here it's nicely illustrated in the figure. You see that part of the chromosome can be, for instance, amplified, duplicated. They can be deleted. They can be translocated, so they're cut out of the original location of the original chromosome and they're pasted in to another chromosome. Or they can be inverted, so they were cut out of the chromosome, flipped around, and built back in. So these are also sources of genetic diversity, but on a larger scale, not on a single base pair scale. And earlier this year, just about 2 months ago, the tiger genome was published, and what they did is they compared the tiger genome with the cat and the dog genome, and they were actually screening for these large-scale changes in a chromosomal structure. And the circular figure illustrates this, because it shows that those parts that were, for instance, found in the tiger as being one chromosomal entity, belonging together, were not found to be a chromosomal entity in, for instance, cat or dog. So, for instance, the one with the yellow shading, you see that it was split up. So, although it was found as a single entity in tiger, if you compare it to cat or dog, you would actually find the same pieces back on different chromosomes. So this really nicely illustrates that if you compare different species with each other, sometimes there are these chromosomal rearrangements. So this genomic architecture plays a role in there. If you want to study genetics for conservation purposes, it's very important that you have a clear research question. So the first thing you do is probably you have to define what genetic marker is most suitable to use. And a genetic marker is actually just a DNA sequence which can be located in a gene or outside a gene with a known location on a chromosome that can be used to identify individuals or species. And all these different markers that are out there, they have different characteristics. Some of them are very diverse, which means that they're very suitable to study something on a population level, and some of them are not as diverse. For instance, if you would study a specific gene, there are more constraints that the gene retains its functionality, so they're typically a little bit less diverse. That would be interesting if you would be interested, for instance, in the evolutionary relationships between different species or different families. So this is an important thing. You have to pick the marker which is most suitable for answering your question. But what's also important is the prior information, because if you isolate the DNA, you need to select this marker. So you depend on prior information, people that have already studied it. It can also be very suitable to use online databases. For instance, if people have studied a particular gene in a species you're also working on, it may be interesting to use that gene as well. And to a lesser extent, it also depends on the source of DNA, because if you need long pieces of DNA, it's typically quite hard to get that from for instance, from scat samples, or what we call ancient DNA samples, like museum samples, because the DNA is already fragmented in those. Another big advantage by using mitochondrial DNA is also that each cell contains several copies of mitochondria, which means you have several copies of the mitochondrial DNA, so even if you have samples of pretty low quality, you still have a good chance of isolating enough DNA to study it. So what would be the next step? And I will go back to the picture I've shown before. This is a part of our data set of lions. We studied a region of the mitochondrial DNA containing one gene and containing a regulatory region. And by aligning all these DNA sequences together, and we can compare all these different individuals with each other, we can draw a conclusion about how all these different individuals are related to each other. Now, this is not on a population level, this is on a continental scale. We're interested in how, for instance, lions in Senegal are related to lions in Kenya, and how they are related to lions in South Africa. So we sampled different populations throughout the entire geographic range to see how these populations are related to each other. So one way of showing these relationships is with what we call a haplotype network, which is shown here. The big dark gray circles are the individual samples, and the larger a circle is, the more samples are in there. So for instance, some some DNA sequences are found in many different samples, which will yield a larger gray circle, and others are quite unique and only found in 1 or 2 samples. And all these circles are connected to each other, and the distance between the circles indicates the number of changes in the DNA you need to get to the next circle. So it means that circles that are close to each other illustrate populations of lines that are also closely related to each other. And I've tried to illustrate this further by showing different groups. For instance, the Central African lines are the group of circles up there, and the West African lines form their own group. And you can do the same thing, or a similar thing, by creating a phylogenetic tree, which is shown here. So here you really look not really what the difference is between the individuals, but what they have in common. And based on what they have in common, you cluster them together on a specific group. So it means that the individuals that are close together, that are clustered in the same group, they have a more recent common ancestor, so they're genetically closely related to each other. And it actually shows pretty much the same picture as the network I've shown before. The West Central African lines are close together, the Central African lines are close together. And the best is if you can illustrate this on a map. So this is what I've tried. I indicated all the sampling locations on this map, and based on the information we got from our DNA sequences, from our haplotype network, and from our phylogenetic tree, we created these groups. And you see that sometimes the groups overlap a little bit. That's what's happening in Ethiopia. For instance, it's the shaded area we find in the same location. We find different haplotypes. And interestingly, these are haplotypes that are not closely related to each other genetically, which means that these individuals probably separated a long time ago, or these populations separated a long time ago. And now we see that there is admixture again. So we see in that region, we see different haplotypes from very different groups, but they're in the same location. And of course, if you see a map like this, or if you have this information, you can use it to prioritize specific populations, because it really nicely illustrates the diversity within the species. So even if taxonomy says all African lions are the same subspecies, when you look at it on a genetic level, we really see difference between groups. What I've just shown was an example of mitochondrial DNA. You remember there's only one allele, so we look at only one DNA sequence. But if you would look, for instance, at a nuclear genetic marker, you would find something more like this. You would find for each individual you study, you would find 2 alleles, one from one parent and the other one from the other parent. And apart from the fact that you can look at the regions or the point mutations in there, you can also look at heterozygosity. So if we take a closer look at these genotypes I have up there on the screen, there is one location where there is diversity. So you see genotype 1 has a C on both alleles, genotype 2 has a T on both alleles, but for instance genotype 3 has 1 C and 1 T, which means that they're heterozygotes. So this information is something you don't have when you study mitochondrial DNA, but if you study nuclear DNA, this is the added information you have. Now I quickly want to take a look at another genetic marker which is very widely used also in conservation research questions, and it's microsatellites. And the interesting thing about microsatellites is that they're very, very diverse. They're diverse to an extent that you can even identify individuals. It's also a method that's quite commonly used in forensics. So microsatellites, they are kind of a strange DNA sequence, because there are very short repetitive sequences, like I have up here, AT, 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 and you're actually not really interested in the sequence of this marker, you're only interested in the number of repeats. And here it's the same thing, you have two alleles, one from each parent, and what you do is you basically count the number of repeats, and you have the same thing here. You have heterozygotes if the alleles don't have the same size, and if both alleles have the same size, the same number of repeats, it's a homozygote. So these are very fast-changing regions. It's a fast-changing region for actually two reasons. One is that it's not having any function in the DNA as far as we know. So if it changes, there's no selective pressure getting it out. And the other thing is that because of the structure, because of this repeat structure, the enzyme playing a role in the replication of the DNA often makes mistakes. So it often adds an additional repeat or at least one repeat out, which means that the mutation rate for microsatellites is much, much higher than, for instance, the mutation rate for a point mutation in another part of the DNA. So to illustrate this, again, I would like to show a part of our data set on lions, because we did this for the lions as well. In this part, we were not really interested so much in the relationship between all these different populations, but we focused on a few populations and we sampled different individuals in these populations. And our data set looks like this. Again, it's very small, but what you see is that on each row there are the individuals, and each individual has two sizes, which is the two alleles. What you can do then is you can use the frequency of the alleles that are found, and you can use that information to kind of group the individuals together with the clustering method. And there is a well-known program called Structure, which is used quite a lot in conservation biology as well. And what Structure does is it's using the allele frequencies to find these clusters, and the graph shows that the optimum number of clusters it's found is 4, and the bar plot shows all the different individuals, so each bar represents a single individual, and it's using 4 different colors, because we found 4 different clusters explain the pattern in the DNA the best, and it's filling up these bars with colors. So for instance, the first few individuals, they have very clearly blue bars, which means that they're very clearly assigned to this single cluster. And the same is, for instance, with the purple and some of the green and the red ones. But you also find individuals where the bars are maybe half green and half blue, which means you see admixture. Between these clusters, there maybe was some gene flow or some hybridization, so we see admixture, which is relatively common if you have well-connected populations. So here as well, we can illustrate on the map where the different clusters are located, and it looks somewhat like this. So we can find these clusters relatively well back on the map, even though we did not feed the program any information about the location of the samples, and we just included the DNA, the allele sizes and the allele frequencies in there. We find more or less the same groups we find with the mitochondrial DNA, which is very interesting. Now, what you can also do, I already mentioned that microsatellites are very diverse, and you can look also at heterozygosity, for instance. It also shows you, can show you if there is any effect of inbreeding, for instance. And this was a quite nice study where they included a lot of large cats, and they calculated all these things based on microsatellite data sets, like the heterozygosity they observed and the heterozygosity they expected, and the number of alleles they found, and for instance the number of private alleles, so alleles that only found in a specific population. And based on that information, you can get an insight into diversity on a population level or on a subspecies level. So interestingly enough, if you take a closer look at the table, the bottom row is the Florida puma, and you see that the numbers for this individual are really, really low. And we will come back to that in the second lecture, because there was a quite interesting conservation project where they genetically had to rescue the very strongly inbred Florida puma. So it's known that this individual is very strongly inbred. So this is also the type of information you can get out of microsatellite data. Now for the last part, I would like to look a little bit more into the recent developments of this field, and quite a lot is happening. So instead of targeting a specific gene or a specific regulatory region, people are now trying to target the variable positions in the DNA directly. And the graph here shows that even microsatellites and AFLWP, we didn't really spoke about that, but it's another type of genetic marker. They're still quite widely used, but you see a very steep increase in the use of what we call SNPs. And SNPs, single nucleotide polymorphisms, actually it's just the same as a point mutation. And so this is almost the same picture I've shown before for the point mutation, and because it's the change of only a single base pair, we will call it a SNP. And what's interesting about SNPs is they are found throughout the entire genome. And I've mentioned before, if you study the mitochondrial DNA, you only have an insight into the maternal lineage, which can give you some good basic insight, but it's not the complete picture. But SNPs can be found on the mitochondrial DNA, they can also be found in the nuclear DNA, and they also can be found on the Y chromosome, which is interesting because Y chromosome is only in males, and it only shows the male lineage. So by combining all these different type of genetic data, you can get a really nice, clear, and all-round picture. The fact that they can be found in nuclear DNA also means that you can look at things like heterozygosity, which is not possible on the mitochondrial DNA. And there are very many of them. So in June 2012, they counted 53 million SNPs that were officially recorded in humans. So there are a lot of SNP positions in the DNA. Now the reason that people started using SNPs, or why there's this steep incline in the use of SNPs, is that there was quite a bigger change in sequencing techniques. So the techniques that will give you the information about the DNA sequences. And there's a lot of happening there in the last few years. So originally we did traditional sequencing, also called Sanger sequencing, and you would result in a single DNA sequence of your genetic marker of interest, as is shown on the left side. So for each individual is represented by a single row, and you have a chromatogram, which is the output of the sequencing machine, and this chromatogram can be translated into a single DNA sequence. But now if you look into next generation sequencing techniques, what they do there is massive parallel sequencing. So it does not create a single DNA sequence of your region of interest, but it creates a lot of DNA sequences. And this is what you see on the right side. So all the rows you see there are DNA sequences of the same individual, and they're all stacked together. So we have a reference sequence up there, and we use the reference sequence to map all the DNA reads of this individual against, and you have a nice alignment of your DNA reads, but all of the same individual. And you can even zoom out a little bit. You really have a lot of information on a single individual. I already mentioned there was really a big change in the last few years, and this is illustrated also by this graph, On the y-axis, there's the output of the sequencing machines using this next generation sequencing. And you see the steep incline during the years, and it's a logarithmic scale. So even though it seems to level off a little bit now, there's still a really big, the output still goes up very rapidly. Now, the main thing why we want to do it is actually illustrated in the next table, and there's quite a lot of information in the next table, but there are only a few columns and actually one single row that are of real importance. Because this table, it illustrates the different sequencing techniques. So the row or the column on the right side, this is the traditional sequencing. And if you look at the different characteristics of the sequencing, you see you get 400 to 900 base pairs with a really nice long piece of DNA, and you get a single DNA read, I will call it for now, a single piece of DNA as an output. So this is the traditional way we used to do sequencing. Now, if we look at one of the next generation sequencing methods that is really frequently used, for instance for genome sequencing, for whole genome sequencing, which is the Illumina, you see that the DNA reads are typically a little bit shorter. They're 50 to 250 base pairs long. But if you look at the number of reads per run, it's up to 3 billion. So your output is huge if you do a run on a machine like that. Now, maybe you're not interested in 3 billion reads of a single individual, but what you can do is you can include several individuals or several regions in a single run, and as long as you give them a unique tag and you can recognize them later while you do your bioinformatics, you can get really a lot of information out. And with keeping that in mind, and you look at the costs of doing this, it's really interesting that if you would say, I want to sequence 1 million base pairs, which is quite a lot if you study a single gene, but it's not that much if you are looking at whole genomes. For traditional sequencing, you would have to spend almost $2,500. But if you do this on a next generation sequencing platform, like for instance Illumina, it's going to cost you 5 to 15 cents. So there's a huge possibility and huge potential in using these next generation sequencing techniques if you set up your research in such a way that you can make use of these new techniques. I already used the example of our line research twice, once for mitochondrial DNA and once for microsatellites. But we were also really interested in using these next generation sequencing techniques. And what we did is we started a SNP discovery for Lion. So we are interested in developing SNPs that are informative for lion populations. We want to find SNPs that show, for instance, that a lion is from West Africa, or a lion is from Kenya or from South Africa. So what we did is we sequenced on the Illumina sequencer, and we included 10 lions and 1 leopard. The leopard is the outgroup, so we have, when we construct our phylogenetic tree, we can use that as the outgroup for that. And all these individuals, they were individually tagged, so we can recognize them when we do our downstream analysis. And luckily, the tiger genome was published, so we can use that as a reference. So again, you see there's a reference on the top, and we have all the DNA reads mapped back to the reference. Now, when you're working in conservation biology, you often don't work with model organisms, so you often have the problem that there is no really suitable reference sequence available. So even if you have no suitable reference sequence available, you can use that technique, but instead of mapping all your DNA reads back to the reference, what you do is what we call a de novo assembly. You just use the similarities of all your individual reads to kind of stick them together in a pretty artificial way, but it gives you a more or less a synthetic reference you can use later on. So in this figure, for instance, we included DNA reads of 3 individuals, a green, a blue, and a yellow individual. And based on the similarities between these DNA reads, we stick them back together, ending up in one red DNA sequence, which we call the contig, and then the contig you can use as a reference for analyzing all your individual samples. So even if you don't have a suitable reference sequence available, this is not the end of the world, you can still use it. The only drawback is that doing a de novo assembly is very intensive for the computer, so you need a very good computer structure, computer cluster to run these kind of analysis on. To give an idea about the amount of data you get from doing this, I mentioned we did 3 runs with 10 lions and one leopard. And this resulted in almost 4.5 million DNA reads of 100 base pairs. So this is quite a lot of information. And assuming that all of these DNA reads are equally divided over our 10 individuals, we can make a really nice comparison between these individuals, and we can use these data to extract the SNP positions. We can directly target the variable positions in there. So I just got an update from our partners in Leiden who do a part of the bioinformatics work for us. They had identified 7,000 SNPs in the first filtering, but I think we can still improve a little bit on that. So I asked them to start a second filtering, and unfortunately this filtering is still running because it takes several weeks for the computer to do all the calculations. But we expect to find more than 10,000 SNPs based on these 10 individual lions. And we can use those SNPs to make a panel, for instance. Based on our 10,000 SNPs, we make a selection of maybe 500 SNPs, and we use those 500 SNPs to genotype more individuals. And because I mentioned you can also look at things like heterozygosity, you can also really zoom in on a population level. And one of the things we really want to do is all the populations we have microsatellite data for now, we want to analyze the same populations by using the SNPs. And our institute has a nice set of machines to do that, where you can do very cost effectively, you can genotype your individuals. So the problem here is the startup costs, the investment you make into developing these SNPs, but once you have them, you can score them relatively easily and cost effectively. Now, so I wanna end off with kind of summarizing that I think when we look at the recent developments in this field, and we look at the SNPs in particular, this has huge potential because this is a genetic marker which gives you the information to reconstruct phylogenetic trees or networks. So it gives you information about large-scale evolutionary forces, but it's also interesting to study separate populations, so it can also give you information on the population level. And if you compare it to sequencing, for instance, a single gene, or you compare it to, for instance, analyzing 20 microsatellites, which is kind of labor intensive, I think this is really a very powerful method, mainly basically because there are so many SNPs to be found in the genome. So I think this is a big promise for the future, and slowly more and more non-model species that are very interesting for conservation research are added for these kind of projects. And I hope that this will really make it possible to gather a lot of information we can use in the field of conservation as well.